Okay. Hello, everyone. My name is Stephanie Hernandez. I have created a short presentation on a very important and broad subject. I have called this presentation Understanding DNA Structure and Replication. So here I have provided what I believe are some of the most important objectives. Number one, the significance of DNA. We will be talking about the crucial role of DNA in genetics inheriting. Number two is the DNA structure. This is the components of a DNA molecule, including nucleotides, sugar phosphate backbone, and nitrogenous bases. Number three, we have the double helix structure. Here we will discuss how the two strands of DNA wind around each other to form the double helix. Number four, DNA replication. DNA replication process. I will introduce the importance of DNA replication for cell division and genetic continuity. Let's begin with briefly explaining why DNA in its replication is so important. DNA serves as the repository of genetic information in living organisms. It contains the instructions necessary for the growth, development, functioning, and reproduction of all living things. It is also the vehicle through which genetic traits are passed from one generation to the next. The accurate replication of DNA ensures that offspring inherit a complete and faithful copy of genetic information from their parents. DNA replication is a fundamental process in cell division. Before a cell divides, it must duplicate DNA to ensure that each daughter cell receives an identical set of genetic instructions. This is essential for the growth and maintenance of tissues and organisms. DNA replication is central to evolutionary processes. Mutations which can occur during DNA replication contribute to genetic diversity. Over time, these variations can be subject to natural selection, leading to the evolution of species. This, in this slide, I will continue to explain more of why DNA in its replication is so important. Knowledge of DNA structure and replication is crucial for medical research and applications. It underlines advancements in genetic testing diagnosis and the development of treatments for genetic disorders. Understanding DNA replication errors is also relevant to cancer research. Next, we have DNA fingerprinting and profiling. This is essential tools in forensic science. The unique DNA sequence of individuals allows for the identification of individuals from biological samples, such as hair, blood, or saliva, even in very small quantities. Advances in DNA technology enable scientists to manipulate genetic material for various purposes. This includes the development of genetically modified organisms, gene therapy, and the production of pharmaceuticals using recombinant DNA technology. Many diseases, including genetic disorders and certain types of cancer, are associated with abnormalities in DNA structure or replication. Research in this area helps uncover the molecular basis of diseases and informs the development of targeted therapies. The few next slides will be to go deep into information and everything that entails the structure of the DNA in its replication process. Let us start with nucleotide composition. An important detail here is that DNA is composed of nucleotides, each consistent of a phosphate group, a deoxyribonucleic sugar molecule, and one of four nitrogenous bases: adenine, thymine, cytosine, and guanine. Also, the specific sequence of these bases encodes genetic information. Next, we have pairing and complementary strands. Adenine pairs with thymine, and cytosine pairs with guanine through hydrogen bonds forming complementary base pairs. An important detail is that this complementary pairing is crucial for the faithful replication of DNA during cell division. Double helix structure. The two DNA strands wind around each other to form a double helix. This structure provides stability and protects genetic information from external influences. This slide is a continuation of more very critical and important information about the structure of DNA. Number one, anti-parallel arrangement. The two strands run in opposite directions, creating an anti-parallel arrangement. This arrangement is vital for the precise reading and replication of genetic information. Role of histones. The packaging of DNA into chromatin regulates access to genetic information during various cellular processes. One important detail is that DNA wraps around histone proteins to form nucleosomes, aiding the compacting of genetic material. Last, we have DNA supercooling. Supercooling plays a role in gene regulation and the dynamic nature of DNA. Also, DNA can undergo supercooling, a twisting or winding of the double helix, to further contact genetic material. These next couple of slides are, in my opinion, one of my favorite parts of this entire amazing process, and that is DNA duplication process. We are going to start with initiation, of course. Replication begins at a specific sites called origins of replication. Initiator proteins bind to these sites and start the unwinding of the double helix. Next, unwinding in helicases. This creates a replication fork, providing access to the replication machinery. Enzymes called helicases unwind and separate the DNA strands by breaking hydrogen bonds between base pairs. Priming and primase. Primates add short RNA primers to the exposed single-stranded DNA. These primers provide starting points for DNA synthesis. DNA synthesis. The leading strand is synthesized continuously while the lagging strand is synthesized discontinuously in Okazaki fragments. DNA polymerases catalyze the addition of complementary nucleotides to the exposed single-stranded DNA. Next up is proofreading and repair. After synthesis, RNA primers are removed and gaps are sealed by DNA ligands. DNA polymerases have proofreading functions to correct errors in nucleotide incorporation. Next is termination. DNA replication is completed when the entire DNA molecule is copied. The result is two identical DNA molecules, each containing one original and one newly synthesized strand. Semi-conservative nature. This ensures the fidelity of genetic information during cell division. DNA replication is semi-conservative, meaning each daughter DNA molecule contains one strand from the original DNA and one newly synthesized. Role of telomeres. Telomeres shortening is associated with aging and cellular senescence. Telomeres, repetitive DNA sequence at the ends of chromosomes, play a role in protecting genetic material during replication. In this slide, we will be explaining another one of our objectives, the double helix structure. Let's start with basic configuration. DNA has a unique structure known as the double helix, resembling a spiral staircase. Picture DNA as a twisted ladder, with each side representing a chain of chemical building blocks called nucleotides. Complementary base pairing. The steps of the ladder are made up of pair of molecules called bases, just like puzzle pieces. Adenine always pairs with thymine, and cytosine always pairs with guanine, like we have mentioned before. Next, we have sugar phosphate backbone. The rails of the ladder are made up of alternating sugar and phosphate molecules. Major and minor groups. The spaces between the steps of the ladder are called groups. Think of these groups as the valleys between hills. They play a role in interactions with other molecules. Okay, let us move on and learn more about that double helix structure. Stabilizing forces. These bonds provide stability, ensuring that the DNA structure stays intact. Hydrogen bonds, like tiny magnets, hold the base pairs together. Role in genetic information storage. The order of the bases along the strands is like a code that carries instructions for building and maintaining living organisms. DNA's double helix structure stores genetic information. Next, we have dynamic nature. This dynamic nature is essential for processes like DNA replication and transcription, where the DNA temporarily opens to allow necessary functions. DNA dynamic and can unwind and rewind. DNA is dynamic and can unwind and rewind. Technological advances. Imagine scientists as detectives using powerful tools to zoom in and understand the intricate details of the DNA double helix. Scientists use advanced tools like microscopes to study DNA structures. Here is a slide with some research and resources available to find lots of reading and information about this subject. I have a couple of journals here. One is the structure of the DNA, and then the other one is called DNA replication, mechanisms and regulations in eukaryotic cells. A couple of textbooks we have here are the molecular biology of the cell. And then we also have another one that's called linear principles of biochemistry. Here are some examples where DNA and its replication is very useful. The Human Genome Project. I'm sure some of you guys have heard about this. The Human Genome Project mapped the entire human genome, revealing the sequence of DNA base pairs. Next, we have DNA replication errors and diseases. Errors in DNA replication can lead to mutations and are associated with genetic disorders and cancers. Okay, so we also have genetic engineering. Genetic engineering techniques such as CRISPR, Cas9 enable precise modifications of DNA sequences. And of course, we all have heard of forensic DNA analysis. DNA fingerprinting is used in forensic science to identify individuals based on their unique DNA profiles. And last but not least, here are a few online resources. Khan Academy has a very good one that's called DNA Structure and Replication. Then we also have Nature Education, DNA Structure and Replication. And last, we have Cold Spring Harbor Laboratory, DNA Learning Center. We have reached the end of this short presentation. I hope everybody liked it and learned a little bit more about this awesome process of DNA replication. If anyone has questions, please stop by the portal and leave them for me. Here I have two more slides with references of where you guys can see all the information that I talked about. Thank you, everybody. Hope everybody have a good day.